Hello everyone and welcome to our first data analysis class. There are lots of reasons to take this introductory course, but I want to begin by discussing our responsibility as scientists. Over the past decade, scientists have been wrestling with the possibility that many published findings may have a large number of mistakes. The worry is that poorly designed studies and intense pressures to publish eye-catching results and sometimes even misconduct have led to a reproducibility crisis. In a recent survey of 1,500 researchers conducted last year by the journal Nature, 90% agreed that such a crisis existed, with 38% calling it slight and 52% calling it significant. These introspective concerns have fueled a burgeoning reproducibility movement, which we want to be part of, where researchers in psychology, cancer, neuroscience, genetics, ecology, evolution, and other disciplines are developing ways to make science more reliable. One solution is to encourage open science, where researchers share their data so that others can more easily verify their work, and where they can publish in a freely accessible journal so the results aren't locked behind expensive paywalls. So I have the Capitol as our backdrop and the Honest Act as a title for a good reason. The Honest Act is a bill that was passed by the House in the fall of 2017, and the act would restrict the Environmental Protection Agency from developing regulations based only on studies whose methods, materials, software code, and data were open and accessible. This sounds good. This is exactly what science folks have been saying, but there is a catch on the policy side. In practice, it would gratuitously handcuff the EPA and prevent it from considering cases that are necessarily less transparent, including those that are confidential, with confidential medical records and proprietary information, and that's the crux there. The act would also force the agency to do a lot of costly extra work, whether it's redacting confidential information or asking scientists to dredge up all the data and code from old studies, which is quite problematic, especially when they're in file cabinets from the good old days before online storage. And it wouldn't produce regulations based on more science. It's likely to produce fewer regulations. Nevertheless, we as scientists can no longer afford to stay aloof from policy, and transparency must be our creed, and that's something we're going to model in this class. In this class, we're going to make the case for open code and open software, thereby increasing transparency. And so the Honest Act, like it or not, at least gives us a framework for discussion. So, next, I thought we'd take a look at some of the results that I alluded to before in that Nature survey. Science is uniquely effective tool for understanding the world, and ecologists and evolutionary biologists have built a robust body of scientific knowledge over the past century. However, several common practices are limiting progress in these fields. For science to progress, results and clear explanations of methods must be shared with all other scientists and the public. Although this fundamental principle is widely understood, there are practices that cloud transparency of methods and results, such as selective reporting, and they appear to be more common than they should. So this is unlikely to be an issue of deliberate dishonesty, which we assume to be really rare in ecology and evolution, environmental in general. Instead, we scientists believe that the unintended negative consequences of insufficient transparency are often unrecognized by many members of the scientific community. So we're going to focus on a particular workflow in this course. This workflow of ecological scientists is currently undergoing a bit of a quiet revolution. Ecological and geographical field data are becoming widely available at an unprecedented rate, and recent years have witnessed a strong push towards a more open sharing of research data. The wide availability of data has also pushed scientists to focus more intensively on the process of data analysis. So data on much of the scientific literature data on how much of the scientific literature is reproducible is rare and generally bleak. The best known analyses from, say, psychology and cancer biology found rates of around 40 and 10% respectively. One recent survey showed respondents were more optimistic, however. 73% felt that at least half of the papers in their field can be trusted. And people in the more traditional physical sciences, such as physicists and chemists, generally show more confidence in this area. So science thrives on reproducibility. And there are two ingredients that are essential for reproducibility in these fields: full disclosure of the methods used to obtain and analyze data, availability of that data, and the availability of how that data was analyzed. This graph is from another recent paper that was demonstrating how their particular lab has increased reproducibility and lowering effort by generally focusing on open source initiatives. Our focus will be data science. For example, data organization, data wrangling, coding, versioning, and documentation, and we're going to be using open science data sources. As you know, we will be using R as the main language for all data kind of preparation and modeling. We're also going to be using RStudio and another GUI graphical user interface called RCommander. In other courses or work, we may use Git or GitHub, which is seen in this diagram as well, for version control, project management, and collaboration. I'm not sure we'll get to it in this class, but certainly in other classes, very likely. We're also going to focus on a positive trajectory towards better science in less time by improving our code with principles of tidy data, something that we'll cover in an upcoming lesson. Standardizing file and data structure and focusing more on communication, in part by creating something called R Markdown documents that will be able to share our code. So why R? R is freely available, open source computing for statistical programming and data visualization. It's got tremendous advantages for research and dissemination of findings. As an open source program, the code for all functions is available. This means that we don't have to blindly trust the software is working as intended. No more black box. Rather, users, us, can look beneath the hood a bit if they choose to do so. I'm not going to focus on that in this course, but you are free to do that as well. There are countless packages that have been developed for R. There are over 10,000 in its repository called CRAM, which are freely available, help making complex analyses with specific context much more feasible. We're going to make wide use of a lot of these packages. Further, R lends itself to reproducible research, meaning that others can view all the procedures taken in any analysis and reproduce those results exactly. It's something we really want to strive for. Reproducible research facilitates transparency and development of research communities with interested users sharing all of our analytical procedures and outcomes. Readers of reports also do not have to guess at any intermediary steps taken during the analysis. If the source code is available, that makes everything more transparent. Finally, the last kind of plug for R is it's immensely flexible. Data is the currency of science, and so we're often going to be asking ourselves, why are we collecting data? It's the currency not only of science, but also of policy, of management, and kind of economic well-being. So I want us to start thinking about why we are collecting data. Are we looking at it for management strategies, which is going to often be the case in this particular course and our focus? We might be focusing on data collection to look at a species that is threatened and we need to protect its habitat. Or maybe we're going to be prepping for the Endangered Species Act. Or maybe we just want to look at some the effects of plant adaptation due to climate change. But all of our data collection should be a systematic acquisition of information, preferably over time, if possible, to support management decisions. And this process is often called monitoring. We want to determine if management objectives are being met. We want to assess the status of, say, populations or habitats being managed. We want to reduce the uncertainty that might be impeding a management decision. So these are some of the reasons why we're going to be looking at collecting data. And ecosystem science is going to be our focus of much of the data in this class. So a lot of the data that I present could be from a biogeography study, botany studies, certainly evolutionary studies, population dynamics, biogeochemistry, and ecophysiology I have circled because these are going to be the kind of primary focuses as well where we're going to get our data. And we're also going to be worried about targeted monitoring. Where are we getting our data from, and how is it going to be analyzed? So these are all things that we're going to be trying to integrate over the duration of the course. So targeted monitoring and sampling design is going to be our next lectures. But in the meantime, I want you to go to step 1 online, get it downloaded, get it running. And I think that's going to be it for this particular lecture. I hope everybody is up to snuff and eager to get going. I know I am, and I'll see you online and in the next lecture. Take care. Bye-bye.